So, Introductory Neuroscience and Neuroinstrumentation, Event-Related Potentials, Lecture 2. So, we went through a few ERPs in our last lecture, and the question arises, why are they important? Okay, we talked about monitoring the auditory, the visual system, and during surgery, but also cognition. So, we can monitor cognitive functions in brain disorders, like for example stroke or Alzheimer's disease, etc. Typically, to do cognitive testing, you need a clinical psychologist who runs a battery of tests, and you can't do it in one session. There are multiple sessions, so 45 minutes, 1 hour each, and there are just not enough clinical psychologists in India or in the world to do this. So we need instrumentation and clinical neurophysiology and stuff to supplement mental health professionals. The other thing is therapeutic interventions. You get, for example, a lot of children are malnourished, not so much anymore in India, but in Africa and other countries. And a lot of companies, have interventions, nutritional interventions, which they say makes your brain better. But there is no way to monitor them objectively except through psychological testing, which, as I mentioned, is difficult because of a paucity of trained people, or ERPs. And then you have newer things, marketing strategies, which suggest that this organic compound, that plant compound, that hamburger is good. It augments cognition. So to evaluate it stringently, we need objective metrics, and ERPs are objective. You can't sham through an ERP. You can't, suppose you ask a person, can you hear this? He can hear it and he might say, no, I can't, and pretend to be deaf. But if you have your P1, N1, P2 complex, you know that the brain is responding to the stimuli. Okay? So these kinds of objective evaluations is superior to subjective evaluations by mental health professionals, typically psychologists or psychiatrists. So that's why they're important. Also, they're quick, they're easy to do, they're non-invasive, it's not such a big deal. Okay? And I have to mention one other thing, that in India especially, there is a huge social stigma to go to a psychologist, a psychiatrist. It is real. People, especially outside the metros, they will not consider marrying into your family if they know that somebody is going to a psychiatrist. It's very real. So ERPs are objective, and they tend, there's no social stigma attached to them. Okay. So let's talk about a cognitive event-related potential called the mismatch negativity. So consider a stimulus, beep, 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 boop, beep, 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 boop. So these are the beeps, and this is the boop. These are the beeps, and this is the boop. So what we do is we average all the beeps, we average all the boops, and we subtract it. And when you subtract it, you get the mismatch negativity. It's a negative potential, and it shows the mismatch signal. Now, when I say beep and boop, I'm changing the frequency, but any change in auditory dimension evokes it, like frequency or duration or timbre. So for example, you have beep, beep, beep on a guitar, and then you have beep on a piano, you'll get the mismatch negativity. So it's pretty much involuntary, and it's an index of central auditory processing. It kind of reflects auditory short-term memory or auditory echoic memory. This is the memory. If I call you and I'd give you a phone number and you don't have a pencil, you just repeat it in your mind, go grab a pencil, paper, and you write it down. Now if I ask you a day later what it was, you would not remember it. It was very transitory. So it's kind of auditory, sensory auditory echoic memory, that kind of memory. Also, it is the only event-related potential present at birth, and hence it's evolutionarily significant. Why? The baby has to recognize the mother's voice. Babies who don't recognize their mother's voice probably have died out in evolution. So it's very important. And the other thing is, most mental health dysfunction like schizophrenia, bipolar disorder, learning disabilities, MMN is reduced. So it's sensitive but not selective. For those of you from medical background, it's something like a blood test where you have sensitivity but no selectivity. Okay. Like erythrocyte sedimentation rate, ESR. It's very sensitive. It goes up if there's infection, it goes up if there's chronic disability, but it doesn't tell you which chronic disability or which infection. Okay. So here it's positive up, which is pretty unusual, and this is your normal beep, which just generates the auditory evoked potential. This is the boop, okay, and these are average responses, not single responses, because single response would be too noisy to see any of this. And when you subtract it, you get the MMN, and the MMN usually occurs somewhere between 150 to 300 milliseconds in that zone. Okay, and which parts of the brain are involved in MMN? So consider a section like this going right like this, and you're looking at it, the brain from the top. So both the auditory cortices on both sides are activated, but there is also another additional area which is activated, which senses the mismatch signal, and that is in the right dorsolateral prefrontal cortex, depicted by the yellow arrow over here. The red arrows depict the auditory evoked potential, okay, areas. So that was the mismatch. Now if you look at PubMed, and if you put in mismatch, you'd get more than 5,000 references. It's been studied very intensively, and it was actually first described by Professor Risto Natanen from Finland. Okay. Another evoked potential, which is even more popular and has got more than 10,000 references on PubMed, is the P300. So the P300 indexes novelty, like you're surprised by something, something unusual in the environment. It's called an oddball, a stimulus. So that evokes the P300. And this occurs a little later than the mismatch negativity, about 300 to 450, 500 milliseconds. It is very constant in a single subject, in trials using a single subject, but between people there could be variations. So you have two stimuli in the typical P300 experimental paradigm, a common stimulus and a rare stimulus which comes randomly, and the rare stimulus usually has a probability of 10 to 20%. Okay. So even small variations, and now we are talking about the visual P300, even small variations can give the P300. So here you have a canonical P300, negative is up, that's a neurophysiological tradition, and you have the P1, N1, P2 response, and this is the P300 response. This is not an individual response, this is average response, probably grand averages from many runs and many subjects. Now I did this experiment in my lab where I used the common stimulus as the E, and the rare stimulus is the buddy E. And you see with the common stimulus you have this response, and the buddy E you have a massive P300 response. And the only difference between these two stimuli is this thing on top, the little matra on top. Okay, so even small variations give rise to this. So we can extend the experiment by, instead of having just a rare and a common stimulus, which is the two stimulus P300 task which you saw before, we can have a three stimulus task where you have this is a two stimulus, you have the standard response which is supposed to ignore, and then you have the target which you are supposed to press the key. So the standard stimulus is usually a small blue ball, the target is a big blue ball, and this comes at a probability of 80, and the target comes at a probability of 10%. But the remaining 10% you have a checkerboard which suddenly pops up, okay, and which you're not supposed to react to. So coming to what it all means. The standard stimulus obviously is the visual evoked potential. So you get a visual evoked potential of that is this guy, okay. Now the target, which is the infrequent blue ball, that reflects working memory, and you get this response which is a P3B, okay. Because you have to keep it in mind, there's a small ball, there's a big ball, when the big ball comes I have to press the button, so this is working memory. The checkerboard on the other hand is a distractor, it's unexpected and unusual, so it reflects attention, are you paying attention or not, and you can actually use these metrics to evaluate a person's short-term memory, his attention, besides the visual evoked potential. So the P3A comes a little early, the P3B comes a little later, okay. Okay, and this dissociation with 3 stimuli allows us to look at 3 things: attention, working memory, and the visual evoked potential pathways. A thing to bear in mind is both the MMN and the P300, they also work for other modalities. Like you can have a P300 paradigm using auditory stimuli, which is what we do at Institute of Science. You can have an MMN with visual stimuli. You can have MMN with somatosensory stimuli. For example, you keep touching, a touch, and then poke. So the difference between the touch and the poke is your somatosensory MMN. Okay. So which parts of the brain is the MMN produced? So please ignore left, top, bottom, and just look at the extreme right figures of the MRI. The P3A, the attention, comes from the dorsolateral prefrontal cortex. The P3B, which is working memory, comes from behind the parietal cortex. And you can actually calculate the dipole. These can be assumed to be dipoles, electromagnetic dipoles, and these green spheres show the dipoles, calculated dipoles. There is no actual dipole over there. The cells, when they act together, they give rise to a field which looks like a dipole. This is the other views of the brain: the top, the bottom, the left, right. But the most important thing to take home is that the P3A attention occurs in front, dorsolateral prefrontal cortex. The P3B occurs in the parietal areas. Okay. So recently, the dream is to use these potentials to see if there's some problem, or to predict, or to scan, or to screen patients with future problems. And recently, the P3A attention and the DMMN. DMMN is duration MMN. FMMN is frequency MMN. Like frequency MMN is beep, 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 boop, beep, 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 boop. DMMN is beep, 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 beep. So duration MMN. So if you combine these two, it seems that you can predict subjects at risk for future psychosis. And this is fairly recent, and but independent groups have confirmed it. So if you can make a device, and you guys are interested in neuroscience and with an engineering background, a dedicated device to record and evaluate P3A and DMMN, well, you have a screening device to quickly go through large sections of population and separate them out. If they have a propensity for this, then you send them to the psychiatrist and the clinical psychologist. But this would be able to be done at the village level by an ASHA worker. Okay. So coming to AEPs and MMN recording from infants. Why do we need record from infants? Because catch them young, as I say. If you can catch an infant with a hearing deficit immediately after birth, most probably you should be able to treat it, because lots of them, for example, have treatable causes like a thyroid deficiency. So all you need is thyroid supplementation, and the baby is fine and grows up to be fine. If this is delayed by a few months, then it becomes permanent, the deficits. And the longer you delay, the more difficult it is to reverse it. So if you can, catch it when the baby is born, like within 48 hours and stuff, we can start thyroid supplementation and prevent congenital deafness due to thyroid deficiency. So this is just one example. There are many such conditions. However, it's difficult to study infant cognition because it's technically difficult. The data is very noisy because the babies just come out, and everything is just kind of settling in. There's a lot of variability, and in India we don't have normative data. So each time you do something, you have to do the normative data a bit first. Infants can't follow instructions. They can't even hold their head straight, keep dangling, and there is no speech. And you can't get behavioral response for statistical criteria, what you can do with children or with adults. You can't, because you're not sure. So there are a lot of challenges in studying infant cognition. Please keep that in mind. But it can be done. So these are recordings done by my group at St. John's, the St. John's Medical College Hospital, NICU, neonatal ICU. So you have a stimulating computer. You have this, this is the recording computer, this is the stimulating computer, and this is the InnoBio, which I showed in a previous lecture, where we're using just the electrodes, and this is the Wi-Fi device, which communicates with this computer. And this is typically a placement. It's difficult to place electrodes on an infant's scalp because you have the anterior fontanelle, that's the bones haven't fused, so it's a little dicey. But forehead is fine. The forehead is just fine, and the recordings would be a little distorted, but we can work around it. So over here, you have data from a newborn. This is the baby was 36 hours old, and remember the DA stimulus from Nina Cross's lab. So this is DA stimuli were given over here at 3 times a second. So it's about 300 milliseconds you get this DA stimulus. And here you have clicks. And notice that within, even though the baby is just 36 hours old, there is a clear difference it can recognize between DA and clicks. Okay. Here the polarity is positive up. Okay. So it can be done. So you can make a headband, abstract all this, and we can record, ERPs from the newborn. Okay. There are some technical challenges. They'll cry, this, that. So typically you have a period of 20 minutes or 15 minutes between feeds. When the baby is fed by the mother, you have 15 minutes to do the recording, and then that's your interval. But that's enough. Okay. Okay. Coming to a slightly older age group. Here, if you remember, a stimulus where even a slight change in the stimulus properties, and if you're using linguistic stimuli, you have a huge P300 response. So the hypothesis was this: can we use Indian language stimuli to increase the power, the analytical power of ERPs in Indian children? Obviously, we can't use it in non-Indians because they don't know Indian languages. So most Indian languages are learnt in an order of increasing complexity. First you learn the vowels A, I, E, then you learn the consonants K, K, G, G, and then consonants plus matras Ka, Ki, Ku, etc. And finally the conjuncts Ksha, Tra, Pra, etc. So we went to the field because we can do it because this is the whole lab, and it's just 2 computers and MATLAB over there and stimuli and stuff. This can be easily used in the field to record data. So we took it to the field, went to Chhattisgarh, and we recorded from first standard children in a government school in Ramanujganj block in Balrampur in Chhattisgarh. And this is GRF from my lab, Shilpa, who helped with collecting the data. And this is a first standard kid looking at this stimuli being presented by this computer. Okay. Why did we use first standard? First standard is where they first get exposed to language, and the argument being that any differences would show up in the beginning. When kids are exposed to something new, kids with problems would really have a problem, which you can see on ERPs, right? So this is the data we got. These were normal active children, and the P1, N1, P2 complex, we call it the LPC, late positive complex. Okay, so normal kids, it occurred at one particular latency of about 425 milliseconds, 10 of these and 10 of these. But in kids who are slow, they're active, everything is fine, they're talking nicely and stuff, but the ERPs were slow, and the teacher told us that these kids were lagging behind. You see a huge difference. Their LPCs are at nearly 670 milliseconds, and in neurophysiological terms, that's a massive difference. It's a biomarker for normal kids and slow active kids. And the bar graphs show statistically highly significant differences between the two. You can barely see the error bars. Okay? And so it was significantly delayed between slow active and normal active. And this is what we had presented to the Society for Neuroscience. The point being that we can use a simple thing like an ERP to straight away say if a child has learning problems, and what problem it has is the next step, dyslexia, dyscalculia, etc. But we can straight away screen a vast set of children. And why is it important? It's important because there are 130 million primary school children in India, and 10% are estimated to have dyslexia. And we don't have the number of clinical psychologists, psychiatrists to handle this, to even screen, forget about diagnosing. So that's why it's important that engineers like you guys devise things like this. So inexpensive, cost effective, but very reliable and based on hard science to screen our pediatric population. So this is the other experiment we did, where we we have these different groups of visual stimuli, vowels, consonants, consonants plus vowels, and conjuncts. So we noticed that there was an increasing, as the complexity increased, the LPC negativity also increased. I'm sorry, the LPC positivity also increased. Here it's negative up, and this is a CZ electrode. Okay. So between this and this, we have a way to monitor, first of all, if the child has a problem, and secondly, where is the problem? Is the problem with the conjuncts, or is the problem really basic with the vowels itself? And depending on what data you get, the teacher can focus on the deficit and make sure, give some extra time to the child and make sure the child overcomes this challenge. Okay. So these are the next experiments we want to do. We want to do semantic incongruity, or called the N400, so negativity at 400 milliseconds. So consider a sentence: the pizza was too hot to eat. That's just fine, okay? Now if you say the pizza was too hot to drink, you have at N400 you have this dotted line negativity happening. Now if it's really weird and incongruous, the pizza was too hot to cry, you have a huge N400. Now this could be applied to middle schools, where you have sentences in Hindi, in Kannada, in Tamil, in Malayalam, where the last word is off, okay? And quickly, again, there's no question of shamming, there's no question of malingering. These are involuntary potentials. Put the cap on, flash all these sentences to the child, and find out if there is a problem, with the N400 or not. If the N400 is there, that means the child can distinguish between obvious grammar issues in the sentence. If it's not there, then the child has to be investigated further. Again, all these ERP based tests can be done by minimally trained mental health workers. Okay. Once the screening is done by them, then they go to the next level, to NIMHANS, to St. John's, to AIMS, what have you. Okay. So we talked about different kinds of cognitive potentials, and talked about how they could be actually used in clinical medicine for societal good, so on and so forth. But what about analyzing? So we talked about ERPs, how you have a time lock stimulus, you epoch it, you filter it, you average it, after artifact rejection you average it. There is another way to denoise the data called wavelet analysis. So wavelet is a kind of frequency analysis which takes off where FFT's Fourier analysis leaves off. And this can reduce recording time significantly, and you can do pattern recognition in large data sets, and the implication is you can do faster clinical tests. And one of the pioneers in application of this to EEG analysis is Rodrigo Quiroga, and he did some very important work in the early part of the century, 2005 to 2008, where he nailed this down. So this is a typical P300 response, which is not average, which is full of noise. The artifacts are there, and after the wavelets analysis is done, you have different components. You can take off components which are not useful, and you can get the P300. And this is amazing because you see it on a single sweep. Normally you have to average 100, but here you see it on a single sweep. And again, this is from Quiroga and Garcia, 2003. So this is the original ERP. It's a bit noisy. After they do wavelet denoising, you see a nice smooth response with the P1, N1, P2, so on and so forth. And consider this a zipped file. If you unzip it, you have all these trials from 0 to 30. Each one is a single trial. There is some interpolation, but it's a bit noisy. It's all. If you do wavelet analysis, it's straight away the noise disappears, and you can see the P1, you can see the N1, you can see the P300, and you can see how these potentials evolve. So in the beginning, the N200, is or the N1 is not brilliant, but it gets better and better. The P1 seems to be invariant, and the P300, the response to some surprise in the environment, that is also seems invariant. So This improves visualization of both visual, auditory, single trial ERPs, and you can apply this to all ERPs from auditory evoked potential, visual evoked potential, somatosensory evoked potential, to MMN, to P300, to N400, what have you. So besides allowing the calculation of better averages, you can also study systematic or non-systematic variations. And as it's fast and parameter free, it complements conventional ERP analysis, which is this on top. So what is conventional ERP analysis? We look at latencies, how fast this potential occurs. We look at amplitudes, what is the amplitude of this potential. I mean, we have to put a baseline over there, what is an amplitude of this potential. Or we look at area under the curve, but usually we stay with latencies and amplitudes. So this is an alternative way, and as I said, this is the unzipped, this is a zipped file. This is the unzipped version where you can actually see single events, single responses. So applications of this could be many, from looking for patterns, any kind of patterns in large data sets. For example, you have cameras on the border, you want to see, if there are any intruders, P300. And if you can get individual responses, then there's something unusual in the environment which needs further study. Okay. So thank you. So that was the event related potential lecture, and we will have demos where we will show different ERPs which will be uploaded to the website. Thank you very much.